Well, I wanted to welcome you here to Health Coach 7, and this webinar presentation is on SNPs and methylation, and a very interesting topic that we're going to have here with regards to methylation and SNPs. And a lot of people are just kind of wondering, well, what is a SNP and how does that relate to health, and what kind of information do I take out of this? I mean, is it something that I need to be concerned about? And is there a correlation to SNPs and the way I feel every day? And a lot of people will ask these questions. Is a SNP really going to cause a major disease process for me? Do I have to worry about my children and my offspring, based upon the SNPs that I may have or demonstrate? And these are all wonderful questions to be asking, but nothing to really start worrying about. Because as we actually move along in this process of understanding the human genome and how changes in epigenetics can affect the human genome, we're going to find that there are so many things that can positively influence possible genetic susceptibilities and sensitivities. And I want to just say today that many people actually have been really, I guess, really stressed over the fact that when their sequenced genome comes back, they see so many different SNPs that are seeming to be worrisome. And the difficulty is that out in the social media platform and even among practitioners, there's confusion. And the confusion actually sets in motion when people are actually moving to treat SNPs using nutritional movement or various types of conventional platforms to address SNPs. And one of the things I do want to say is you don't want to be basing your treatment on just treating SNPs. I always say go back to the gene because the gene is the authority. And we always need to be going back to the gene to see what the functional control of the gene is. All right. So to begin with, let's identify what a SNP is. Okay, a SNP basically is the most common form of variation there is, and most of the population, as you see with this slide, developed by the National Cancer Institute, the common population demonstrates a large, variety of different SNPs in their human genome. So now, when we're actually looking at SNPs, we have to understand what is a SNP. A SNP is identified as a single nucleotide polymorphism, and simply a SNP is a typographical, I would say, insertion of a letter in the base pair movement that actually replaces one base pair for another. And so we wind up seeing that the SNP, when, like for instance, you see on this slide here, going from G to C in this variation on a particular gene, may be related to some type of expressed sensitivity on that particular gene's functional control. Now, I want to just make it clear to you that SNPs, generally speaking, are non-consequential, which means they do not cause serious disease process, okay? And a lot of the times what we wind up seeing is a SNP is isolated out on a single gene, for instance, like MTHFR, and we wind up making a cardinal point at an MTHFR if we have two positives, for instance, which you'll notice that on your genetic report you'll have a double positive or a positive negative or a negative negative. And these terms respectively, double positive is homozygous positive, positive negative is heterozygous, and double negative is a homozygous negative. Now, what those implications basically are is that the two sides of parental inheritance is contributed to the mutation. So that means that you're getting it from your father and your mother's side if you're positive positive. If you're positive negative, you're getting it from one parent, and if you're negative negative, you're not getting any mutation at all. Okay. Now, what I usually been instructing many people that ask me is that when you look at a SNP combination, should I be isolating those SNPs out alone? And I would say no, you should not be just considering accumulating those SNPs and then trying to see if whether or not there's an express change on the particular pathway that the gene controls. Now, a lot of the time you can look at these SNPs and you can say, well, I can see some functional correlation with, for instance, MTHFR and the ability to methylate appropriately. And you have to remember that SNPs can affect methylation process, that is for sure, okay? However, we always go back to the gene functional control, and we always must consider looking at relationships of cross-related genes. So in other words, there are sister genes, if you will, that actually will complement or cause another variation on performance on a gene, all right, if it has a similar type of a control in a certain type of function of it, like for instance, looking at mitochondrial function of a cell. Now, the mitochondria of a cell is the powerhouse, and there's what they call an electron transfer gradient that happens there to generate power. And there are many, many things that are dependent upon a efficient mitochondria. And for instance, phosphatidylcholine is one of those interesting fuel molecules, if you will. And phosphatidylethylamine methyltransferase, PEMT, is a gene control that's very important in the development of phosphatidylcholine, which actually produces choline. And it's very important to know that if there are SNP variations that affect MTHFR, and if there are SNP variations that affect PEMT, we're going to find a cross relationship that occurs in the methylation cycle. And both of these genes will be contributing to methylation disturbance then if there's any expressed sensitivity there. Now, as I mentioned earlier, I would say greater than 97% of the time we're going to find that SNPs alone are not responsible for causing disease process. So when we talk about SNPs, we need to be talking about epigenetic shifts as well. The thing that we really need to be focusing in on is creating negative signals in epigenetics to where maybe methylation changes associated with SNP variations may cause, expressed concern for disease patterns if the programming of epigenetics doesn't happen. So, like in other words, epigenetics basically is signal controlling that are responsible for assisting with keeping a balanced methylation pattern so that there would not be any negative signals causing what they call higher sensitivity and susceptibility to developing a disease associated with the human genome sensitivity. So really the important thing to remember here is that SNPs and epigenetics are two very important systems. So if we do see a pattern of SNPs to where they are likely affecting different SNPs affecting performance of genes, we're going to see certain type of patterns in a person's health. For instance, you might actually see progressive autoimmune reactivity. You might see more activity toward inflammatory controls. You might see a slower ability to develop certain type of fuel molecules like phosphatidylcholine to produce choline, which can definitely affect the health of individual cells, as well as working in the central nervous system, which the central nervous system and the peripheral nervous system depend upon choline. You're going to find methylation of the brain would be disturbed by choline disturbances as well. So you're going to see these type of patterns associated with these type of changes. Now, bottom line is when we look at SNPs, what we want to say is if we do see SNPs and cross-related gene relationships with SNPs, we look at epigenetic mechanisms and we say what are the factors that are involved in affecting the methylation cycle. And bottom line is, if you have a lot of single nucleotide polymorphisms that are affecting different gene controls, the basic diagram that you see here is just kind of outlining it really well. So you can see epigenetic mechanisms influencing DNA methylation. Okay, and the bottom line is SNP movement is an integral issue with regards to keeping a DNA methylation cycle regular. Okay, so now you've heard of hypermethylation and hypomethylation. So hyper is over-methylating, hypo is under-methylating. Both have their consequences. But the bottom line is that if we don't have good epigenetic adaptability and intelligence, we are going to have what you see on the right side of the diagram, some pretty more serious type of degenerative disease patterns, including cancer, autoimmune disease, neurodevelopment disorders, mental disorders, diabetes, et cetera, et cetera, et cetera. So bottom line is, it's a SNP. A single SNP is not necessarily going to cause a disease pattern. However, SNPs across the board, which there are 15 million of them in the human genome, and 23andMe sequences about 1 million of those. Okay, so there's a good 14 million that we have no idea what relationships there are. But the ones that we do know, and we can correlate with some good, going back to the gene function relationship, we can see that there might be patterns in related genes with certain type of SNPs affecting certain type of function. So the bottom line is, ultimately, a SNP is not going to cause a consequence of disease alone. A SNP manifested across the related genes that have maybe related functions may be implicated in plotting a higher risk for disease development. But the good news is that epigenetic mechanisms and movement in creating epigenetic intelligence can change those negative patterns that you see that your human genome might plot for those sensitivities and susceptibilities, and promote a better signaling to protect you from the vulnerabilities of that your genome is actually manifesting. Okay, so basically, creating good health platform is to not just to become fanatical about the SNPs that we see. It is about being able to create an epigenetic intelligence platform to address certain type of patterns that we might see that the human genome is actually demonstrating. So now remember, you can't change your genome. There are 23,000 plus genes in the human genome, and that controls about 60 trillion cells, 15 million SNPs with 3 billion base pairs. I mean, we're talking about a phenomenal large, very, very large platform. So as you can see, collectively, we've got a lot of information to obtain to be able to create a better, what they call pathway genomics movement, and giving a better, more precise epigenetic intelligence type of a program. So now what we could do now is we can take our various things that we can do, like dietary factors, exposures, for instance, heavy metal exposures, environmental exposures, the likelihood of being more sensitive to cardiovascular related inflammatory activity, autoimmune related inflammation, gut disturbances, and poor gut brush border management, where you don't have very good protections in the gut, or protection even in various other places in the mucosal surfaces, like the mouth and along your sinuses and your GI tract. Very important to know that these factors in creating epigenetic changes are going to require some very good nurturing inputs to reduce the likelihood of creating bad epigenetic signals. And I hope that makes sense for you. But that's really where we're going with this. We want to be able to see that it's important for us to know that epigenetic patterns can favor, bottom line, healthy cell expression. Here's a very good article here that actually discusses the how genes are methylated, and you can actually research this out yourself. Some of it is in real high scientific language, but I've actually referred to this before, talking about various type of relationships in hypermethylation and hypomethylation, and the relationships that might predispose to certain type of diseases like cancer. It's a very good article to be looking at. This is actually in genome biology a little more sophisticated, and this is actually discussing basically characteristic changes in DNA methylation patterns. As it says here, DNA methylation plays an important regulatory role in eukaryotic genomes. So in other words, in our genome, in more high sophisticated cells, we're going to find that things like the DNA methyltransferases, DNMTs, are responsible for maintaining methylation patterns, and that affects the DNA regeneration and replication in a normal fashion. So if we are going to be looking at SNPs, the more SNPs we develop, there might be some SNPs that are favorable SNPs for adaptation, and there may be SNPs that are unfavorable for creating more risk. So the whole idea is epigenetic intelligence to create more protection against risk. So this is a good article to kind of keep in mind. This is an oncogene journal here. It's talking about DNA methylation, methyltransferases, and cancer relationships. You're going to find that I've actually done a lot of work in cancer, so you can actually see relationships here. There's a lot of information on telomere lengthening and shortening and protection of methylation, healthy methylation of DNA. And then this is a very good simplistic, what is DNA methylation? I would suggest that you spend some time, just perusing that. And a very, very interesting video. It's actually called The Symphony in Your Cells. Really love this video because it actually talks about, in a very distinct way, how the harmony of these cells together with the DNA sequence is very much a collective work, if you will. So in other words, various types of harmony produced by our genetics. For instance, it says here, a large international group of researchers has put together 111 epigenomes from different human cell types, and then the team also assembled epigenomes of certain stem cells, and they came up with some phenomenal relationships. And I think listen to this and watch this video because it's really explains it very nicely. Now, bottom line is we get back down to this diagram that you probably recognize very well, and this is the methylation cycle. So now, when we're talking about SNPs and the insertion of letters where letters should either not be, or maybe for adaptation should be, bottom line is we're actually trying to see what the effect would be on the overall methylation cycle. And as I've mentioned to you, methylation is an integral part of metabolic function. It's a biochemical process. And as you can see here, it's involved in a lot of different exchanges. And when you actually are looking at all these exchanges, bottom line is Krebs cycles an energy wheel responsible for generating energy for your energy metabolism, which includes the nitric oxide synthesis, which is also involved in a lot of detoxification mechanisms. You're going to see down here, we're talking about how glutathione is going to interface with superoxide dismutase. You see in neurotransmitter relationships up in here, methylation effects on the methylene tetrahydrofolate, and here's your neurotransmitter relationships, which you're probably recognizing some of these characteristic gene controls. And then your homocysteine, cystathionine, ammonia, glutathione movement here. So remember, glutathione, even over here, this glutathione is going to be affecting your ability to detoxify and also to protect against pro-oxidative risk. So what we want to do, free radical protection and protection against high oxidation, that might cause more damage to cells and facilitate more inflammatory changes, more harmful changes on the membranes of the cells, and overall affect the systems that they control. So, I mean, it's just very important. And then you see here in this cycling here, the relationship with the DNA and RNA, which is affecting, the ribose and nucleic acids and sugars that are involved in developing healthy DNA and RNA. So bottom line is that SNPs and SNP relationships across the genes. In other words, we just don't want to be looking at just SNPs by themselves. And I'm just going to pull this up. I love to go to gene cards here to just kind of give you an example of that. But I want to give you some solid ground so that you can see that when we're talking about SNP changes and epigenetic controls, we're basically saying we want to be looking at this from the standpoint of relationships. And I'm pulling up MTHFR because it's probably the most widely studied gene SNP profile to show us that what relationships on methylation. But as we look here, when you're actually looking at a gene, you should always be looking at how a gene is named, what it's responsible for, what the gene influence susceptibilities may be, and then what the relationships are, where it's located. As you can see, chromosome 1 there, the size of the number of base pairs that there are there, as well as the number of amino acids that you see, 656 amino acids. And then you always want to go back down to the relationship of the cell. So in the cell itself, this MTHFR is very much seen in the cytosol very abundantly, and even in the extracellular space, so it is a communicating enzyme. But what I really want to show you is that there are relationships that you need to be looking at for cross-related relationships with these genes. And that's why I say when you're looking at SNPs, you don't want to just be looking at SNPs of one isolated gene. You're going to be looking at SNPs of maybe the surrounding genes that may have some relative movement on that gene's performance, either directly or indirectly. And as you can see, MTHFR does not stand alone in this sea of movement. You're going to see SHMT2, which actually which is a very important gene control as well. You're going to see it related to CTH. You're going to see it related to MTR, some other methylene tetrahydrofolate receptors. Okay, for instance, on the SHMT2, you're going to look at that and say, that what is that? That's serine hydroxymethyltransferase, and it contributes to mitochondrial control and biosynthesis, interconversion of serine and glycine, very important in maintaining mitochondrial DNA. So you're going to see that SHMT is very important in regulating methylation cycle in the mitochondria. Okay, so that's why I wanted to show you here that you just don't isolate one single gene and SNP variations that you see in that gene. You have to be looking at cross relationships, and then you might be able to see when you're looking at SNPs with the cross-related genes how they relate to functional health as a whole. So for instance, I've been seeing a lot of people with a lot of mitochondrial disturbance, a lot of cell energetic issues, a lot of protection against inflammatory, various type of genes that seem to have some relationships with each other, and these SNP variations, are present. And people that are experiencing this, even before even looking at their health history, you can pretty much see that these patterns exist. And when you look at the health history, it confirms, wow, these are the symptoms that you're experiencing. A lot of people with associated high-level mental brain fog or generalized fatigue when they're actually showing poor methylating movement, that actually influences, immune and inflammatory pathways. It's just amazing how these things, you can see it, and without even looking at the health history, as I mentioned, I can see some predictive movement. But I always want to come back to, it's about creating epigenetic intelligence to be able to create a better adaptive platform to protect against the susceptibilities and sensitivities that are seen. And remember this, that the genes are always moving, your genetics It is what it is. If you're a black striped zebra with white background, you're not going to be a white striped zebra with a black background. I use that as an analogy. But you can make one powerful, strong black striped zebra if you see susceptibilities that are unfavorable by making those changes that we are talking about. So anyway, I hope you find that this is helpful. And I'm not able to take questions today because I'm pre-recording this. I just want to hope that this would generate some excitement for you so that you could be looking at exploring the relationships in your genome. And that's basically the bottom line, is how can I change my susceptibilities by creating better epigenetic signals? All right, well, it's just great to present this today, and we'll be presenting our webinar next week as well. You can look at the contents that'll be posted, and I look forward to working with you then. If you have any questions whatsoever, you can send some of your questions to info@healthcoach7.com. And if you're interested in talking with me with regards to your genetics and how we can figure out the maze of genetics, you can always set up a consult with me, and I'm happy to work along with you. Anyway, good day, and see you next time.